Right. Now that we have looked at RNA and DNA, we can dive into DNA replication. But just before we dive into DNA replication, let's look at what is the difference between a nucleotide versus a nitrogenous base, so that we don't make the mistake of thinking a nitrogenous base is a nucleotide. A nucleotide is made up of that nitrogenous base, a sugar. In this case, it's a DNA, so it's in DNA, so it will be made up of a deoxyribose sugar and a phosphate. So this image that you're seeing here that I drew is a nucleotide. A nitrogenous base is just this part over here. It's a nitrogenous base, such as, for example, your adenine, thymine, guanine, and cytosine. And remember, we have what we call base pairs. Base pairs is I basically mean that adenine will always pair with adenine, and then they'll be bonded together by those weak hydrogen bonds. And then subsequently, adenine with thymine, thymine with adenine, guanine with cytosine, and cytosine with guanine, whichever one you want. Good. So with that in mind, let's dive in straight into DNA replication. DNA replication. Now, with DNA replication, let's start here. Remember what is replication? A replication is basically making a copy. Basically, for example, if I wanted to go to the photocopy machine to make a copy of my idea, I'm making a copy of my idea, an exact copy to come out the way it is, the way the original is. So, excuse me. With DNA, when the cell, remember from mitosis, when the cell is in interphase, right? Just before the mitotic phase, where before it goes into prophase and then on and on and on up until cytokinesis, before it goes into the division state, it needs to make a copy of the DNA for the next daughter cell, right? The exact same copy. So how does it do that during the synthesis phase, the S phase, in preparation for mitosis? So that's what we're going to be looking at, right? Good. Now, how do we look at it? We're going to be looking at it in steps. I want to look at it in steps. Step number one, remember that DNA, if for example, it's not in interphase, it is coiled up like this. If it's chromosomes. Chromosomes are not found in interphase. So basically, after mitosis happens, they relax, né? But they're still coiled up, né? To form that chromatin. But they're still coiled up, right? Now, around those histones to form nucleosomes. Now, what do they need to do? They need to start unwinding even further. They unwind even further, né? Unwinding first. So the first thing it does is to unwind. Yeah? Unwind. Basically, that's the first thing that it does. So after it unwinds, then let's use these strands over here. I'll use these strands over here as my original DNA. 2, 3, 4. Let's use 4 of them. 2, 3, 4. Remember, these were bonded. So let's use A, G, C. So if it's G, C, the other strand will be T, C. Okay, it's bonded with hydrogen bonds, and then G. So basically, this is our DNA. After it unwinds, now we can see the nitrogenous bases and then the backbone, which is your phosphate and sugar, right? Good. After it unwinds, it needs basically to expose these nitrogenous bases by unzipping. So those weak hydrogen bonds between the nitrogenous bases will therefore unzip. That's step number 2. Now we say it unzip. So it's sort of like having a zip. You unzip it. you expose the two strands. So let's draw it on the side here. So let's say these are the two strands after it unzips. 1, 2, 3, 4. 1, 2, 3, 4. Still has the same nitrogenous bases. That's adenine on this one, adenine, guanine, and cytosine. Then thymine on this one. Remember now, excuse me, the weak hydrogen bonds have been broken, so these are not bonded together. That's thymine again, that's cytosine, and that's guanine right over there. So it has been unzipped. So the two strands are basically available now for replication. Good. Let's name this strand number 1, strand number 2. But what are we going to call them? We are going to call them template strand number 1. So these are called templates. Template. So why do we call them template strands? So these are the strands basically in which new strands will be built upon, right? And what do we call those new strands that are going to be built? We call them complementary strands. Complementary strands. Now, that is us moving now to step number 3. Now, in step number 3, good. Now that we have all of these complementary strands, good. Each original DNA strand serves as a template on which the complement is now going to be built. So how will the complements be built? Remember, let's use this as basically, yes, good. I'll use this the red color. Remember, we have free nucleotides. Free nucleotides around the cell. C, G, C. Like they are all over the place. Let's pretend that they are all over the place. A, T, 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 C, G, G. So now what's going to happen? These free nucleotides, remember, it's not just a nitrogenous base, it's a whole nucleotide. They are going to go and basically bond with the template strands. The free nitrogenous bases on the template strands. So what's going to happen now? They're going to start building a strand now, a new strand on each template strand. So what do we mean by that? Now, let's do this. Different color. Let's redraw those strands. In step number 3, the free nucleotides begin to bind to the template strand. Still is template 1, template 2. Still carries the same code, which is basically your A, G, C, and then T, T, C, G. Great. Now, all of those therefore come, the free ones. Let's say, for example, it will start. This will need a G. This requires a C. So they start building what? They start building new strands. This one will come in here. There will be an A over here, bonding with an adenine, bonding with a thymine, and then another adenine bonding with a thymine, together building a new strand. What type of strand are they building? A complementary strand. Therefore, this takes us to what? This takes us to step 4 now, where basically they all attach and then they form new strand. As this happens now, we're going to end up with this. Let's look at it. We're going to end up with template strand number 1 and then complementary strand. Let's use the black color now. Complementary strand. Template strand number 2, complementary strand number 2. The genetic code is still the same. Still the same. What do we have? Remember, in the original we had adenine, and then guanine and cytosine. And another template, template 2, we had thymine, right? We had thymine and thymine. Let's do it like this. It's actually on the other side. So we had thymine, and then cytosine and guanine. Great. Now what happens? Now remember, we had free nucleotides to bond with the template strands to form a complementary strand. What happened? Now we have adenine bonding with thymine, and then another thymine bonding with adenine, guanine with cytosine, with guanine, and that forms a template strand. Same happens that side. We will have adenine bonding, forming weak hydrogen bonds with thymine. Another adenine, same. Guanine, the same. And then cytosine, the same. Now, do we agree from this given example that we have two new DNA molecules that are exactly the same amount of nucleotides, the same type of DNA, AT, GC, CG, GC, CG. So that is what a cell does during interphase when it makes a copy of itself. Then you end up with what? You end up with two identical two identical nucleic acids, new nucleic acids, which basically are what? Which basically are going to separate during cytokinesis. One goes to the nucleus of that cell, and one remains in the nucleus of the parent cell. So why does replication happen? What is the role of replication? To make sure that the daughter cell has got the same amount of DNA, an identical DNA, same number of chromosomes, for example. Great. So basically, this is DNA replication in a nutshell. In the next lesson, we'll quickly look at errors during DNA replication. What do they cause? And then we'll move on to DNA profiling. But before we go, I have a simulation that simply aids us in basically seeing what really happened. So in summary, what we just did is the following. So we'll have a DNA strand. How do we know that this is a DNA strand? With thymine is available. Thymine is available. So let's bond them together. Now we want to see what happens during replication. Now we have a whole DNA strand. So basically what happens is that with the help of some proteins, for example, first and foremost, it must unzip. It must unzip. Basically they open, they separate. After they separate, now there are two template strands. Two template strands. Template strand number 1 on your left, and template strand number 2 on our right. And then with the help of other enzymes as well, now we can actually come and form new complementary strands on the templates. So how do we do that? Let's start with template number 1. We need free, these are our free nucleotides. A free nucleotide is going to come and bond here. Another one here, adenine with thymine. If we see guanine, that means cytosine is going to come and bond over here. We see cytosine, guanine is going to come and bond over here. Now, can you see? It's forming a complementary strand right next to the template strand. The template strand is the original strand, basically. Right? Great. That happens. And also on template strand number 2, the same thing happens, but in a different direction. Good. We're not going to go into detail about which one goes from where to where, but all you need to know is that a complementary strand will be formed on both of them, and that's how it happens until we have the same DNA on each side. So what happens at the end of the day? At the end of the day, you end up with, let's use a different color. You end up with template strand 1 and complementary strand 1. And then subsequently template 2 with complementary strand number 2. And that is DNA replication in a nutshell.